Dear students, அனைவருக்கும் வணக்கம், வந்தனம், நமஸ்காரம். Today I'd like to give a brief idea regarding additional one mark questions in classical genetics. Classical genetics, the study of heredity and inherited variation, is said to be genetics. Classical genetics, completely based on Mendelian experiments, monohybrid, dihybrid, monohybrid backcross, monohybrid test cross, and other things. So simple questions, additional questions I want to discuss today, students. Question number one, you know pretty well, father of genetics, Gregor Johann Mendel. Our book பேரு உங்களுக்கு தெரியும். Experiments in plant hybridization by Gregor Johann Mendel in the year 1865. அப்படிங்கிறது தெரியும். And the experiment material selected by Mendel for monohybrid and other crosses is Pisum sativa, the garden pea plant. Then in the end of monohybrid cross, Mendel proposed two laws. One is law of dominance and another is law of segregation or law of purity of gametes. Question number 3, the phenotypic and genotypic ratio of monohybrid cross. You know, physical expression of a character is said to be phenotype, and the genetic expression of a character is said to be genotype. The phenotypic ratio of monohybrid cross is 3:1, and the genotypic ratio is 1:2:1. 1 homozygous tall, 2 heterozygous tall, and 1 homozygous dwarf. Next question for you. The test cross ratio of monohybrid cross. You know, test cross, crossing the F1 hybrid with its recessive parent is said to be test cross. The monohybrid test cross ratio is 1:1. The dihybrid test cross ratio 1:1:1:1, 50% parental combination and 50% recombinance due to law of independent assortment. You know, in the end of dihybrid cross, Mendel formulated. In the end of monohybrid cross, Mendel formulated law of dominance and law of segregation. In the end of dihybrid cross, Mendel proposed law of independent assortment. Whenever two different parents with two different characters with 4 different expressions are crossed, the assortment of characters independent to each other. Two different parents with the 2 different characters with 4 different expressions are crossed. The assortment of characters are independent to each other. That's why the new varieties, yellow wrinkled and green rounded, formed in the end of dihybrid cross. The characters selected for monohybrid cross experiment, Mendel found around 34 varieties in pea plant, and he selected 7 varieties for his experiment. Adula monohybrid cross cover the character selected by the Mendel for the monohybrid cross, height of the plant or height of the stem, tall plant with 180 centimeter and dwarf plant with 45 centimeter. The character selected for dihybrid cross by Mendel. Dihybrid cross, a cross made to steady the inheritance of 2 pairs of contrasting characters. One is a seed shape, a round is dominant over wrinkled. Second is cotyledon color. Yellow color is dominant over green. So seed shape and seed shape and cotyledon color selected by Mendel for dihybrid cross. Incomplete dominance experiment conducted by, if the F1 hybrid produces a phenotype which is a mixed expression of both the parents, then it is called incomplete dominance or blending inheritance. So Mendeloda incomplete dominant, Mendeloda law of dominance against either. So incomplete dominance or blending inheritance reported in plant name உங்களுக்கு தெரியும், Colcons mirabilis jalapa னு ஒரு plantல பண்ணார், அது இந்த இடத்துல ஞாபகம் வச்சுக்கோங்க. So incomplete dominance or blending inheritance experiment conducted by Colcons in mirabilis jalapa 4 o'clock plant and snapdragon antirrhinum majus. இதோட ஸ்பெஷாலிட்டி என்னன்னா, in incomplete dominance the phenotypic and genotypic ratio ரெண்டுமே ஒண்ணாதான் இருக்கும். The physical expression as well as genetic expression is 1:2:1. The botanical name of 4 o'clock plant, Mirabilis jalapa. snapdragon சொன்னேன், அதோட botanical name Antirrhinum majus. The phenotypic and genotypic ratio of codominance in Gossypium species, in the Gossypium species, that is codominance. If the F1 hybrid produces the expression of both the parents, we are calling it as codominance. AB blood group, ABO blood group. AB is a very good example for codominance. Both the genes are present in the same organism and the character is expressed. Codominance, Gossypium species number botany, the ratio is 1:2:1. Lethal genes spelling is little genes. This is lethal. The gene which causes the death of the organism is said to be lethal genes. Lethal kill pannuva. Snapdragon la first report panna scientist oda per enna E-bar. This is not a little gene. This is lethal gene. Sorry, typographical error. Check pannikongo. This is lethal abdingatha nyavachko. Next, dominant epistasis. Dominant epistasis ratio is 12:3:1 in cucurbit appe pola banana summer squash. And then the ratios nariya kuduruka adala marandudadu. Recessive epistasis ratio is 9:3:4 in Antirrhinum majus. Duplicate genes with cumulative effect ratio is 9:6:1 in the organism la nyavachko. Complementary gene experiment ratio is 9:7. Supplementary gene ratio is 9:3:4. All ratios start from 3:1, 1:2:1, 1:1, 9:3:3:1, 1:1:1:1. The phenotypic and genotypic ratio 1:2:1. This 12:3:1, 9:3:4, 9:6:1, 9:7, 9:3:4. Or match Mary Kakelam, pick out the correct pair, incorrect pair. Then inhibitor gene ratio 13:3. Duplicate gene ratio 15:1. So all these ratios you should remember. Then heritable variations in organisms due to variation we differ from our parents. If it is inherited to the subsequent generations, we are calling it as heritable variations. If the variations are transmitted to the subsequent generations, we are calling it as heritable variations due to law of independent assortment. The recombinants are new varieties are formed in the end of dihybrid cross. No medal crossed yellow round with a wrinkled green. In the F2 generation, 9:3:1 ratio of yellow round, yellow wrinkled, green round, and green wrinkled seeds are formed in a particular. The recombinant are yellow wrinkled and green round, or the recombinants are new varieties are formed in the end of dihybrid cross. This many questions. Best wishes. Thanks a lot for hearing. Thank you very much. Thank you.